Materials supplied by Microsoft Corporation may be used for internal review, analysis, or research only. Any editing, reproduction, publication, rebroadcast, public showing, internet, or public display is forbidden and may violate copyright law.
Following such a fantastic talk is that you have much of your motivation already completed for you. And Haifeng has done a fantastic job of outlining some of the developments around the growth of sequencing data and some of the potential applications that are now making their way into the research and into the clinical communities. I would go as far as to say that we are in very broad agreement even on the choice of some of our slides, so we'll see that in a moment. So our agenda today is fundamentally about tools, and particularly about the use of tools based on the .NET Bio project. So whilst this is not sort of an introduction to the project because it's been around for a number of years now, it is a bit of a snapshot of where the project sits and some of the work that's being done to extend it and some of the work that's being done to build things on top of it. But for those who haven't seen it, I'll spend a few slides just introducing what the library actually does and some of its history and some of where it might head in the future. Then we'll move on to some increasingly interesting applications, both in terms of its application directly in high-throughput genomics, but also in its application in other contexts, so its extensions into proteomics and also the possibility of integrating it with other languages and also even to utilize it as a backbone for the idea of doing bioinformatics somehow in the browser. So, as Haifeng pointed out very nicely, genomics has kind of come a long way over the last decade or so, and we've particularly agreed on this slide as a way of indicating some of the changes that have taken place over that time. Fundamentally, the .NET Bio project is about dealing with genomic data, and it's about sequence data, whether it's really nucleotide data or proteomic data. But what has accelerated the use of libraries such as .NET Bio and other open source approaches to bioinformatics is the need to deal with very, very large amounts of data, and particularly the need to deal with very, very large comparisons which arise because of the data which has arisen because of next-generation sequencing projects. And what we're going to talk about here is just some of the changes in the nature of the computations that have been brought to bear in recent times. So as we've moved over that time, of course, we've seen not just one sequencing revolution, but a number. And so, as was pointed out before, we're starting to see people moving not just to work based on a small set of sequences, a reference sequence, or a small set of individuals, but rather population studies based upon sequencing of a very, very large number of individuals. So we're starting to see early signs of personalized medicine. Some of the predictions may well have been optimistic, but we're certainly starting to see movement in that direction. And we'll particularly focus here on the notion of genotyping and identification of SNPs based on resequencing projects. So the .NET Bio project itself began a number of years ago as a project initiated from Microsoft Research and Microsoft Research Connections. And it's a language-neutral open source toolkit for bioinformatics based on .NET technologies. The current version is built on top of .NET 4.5. Fundamentally, it allows you to build applications based on any number of CLR-compliant languages, so C#, F#, Iron Python, Iron Ruby, and so on. It's open source, released under the Apache 2.0 license under the auspices of the Outer Curve Foundation, and it's fundamentally a foundational library on which other people can build software and to enable people to make contributions. As one might expect of a genomics, what is fundamentally a genomics library, it provides a class library of standard components for some of the things which have underpinned bioinformatic computation over the last decade. So parsers for particular file formats, support for different types of sequences, fundamental algorithms for search and assembly and alignment, and also an ability to extend its capabilities by linking to external web services through a well set up facility for doing so. So this is just one of the help files giving you a fairly good idea of some of the algorithm namespaces. So we have alignments, we have assemblies, we have a port of mama, a whole bunch of different algorithms and facilities being available for use. So in recent times, the .NET Bio project has grown in acceptance and also in importance amongst people working in the next generation sequencing area, including from some people who are involved in sequencing companies and sequencing startups. The idea of the library, of course, was that it would provide fundamental data structures to encapsulate what we're working with, so fundamentally sequences, fundamentally about parsing, and of course the ability to execute such algorithms relatively quickly by taking advantage of some of the parallel extensions and other access to high-performance computing, which is provided by the .NET libraries. So the library itself is both a collection of functions, a collection of methods that is there to be exploited, but also it comes as a set of pre-assembled or pre-constructed tools to enable people to use things. So there are a number of developed utilities which ship with the library. So these exist for sequence comparison and alignment, so things around MUMA and NUCMA. Also utilities for working with sequence alignment map files and their equivalent BAM files, and also tools for de novo assembly of sequences from short reads. So that's a thing called PADINA. One of the other aspects of the library which has caused it to get some interest from educational institutions in particular is the integration of these tools with office tools such as Excel, which makes them very useful for undergraduate bioinformatics classes. And so we've actually written some material to work in undergraduate bioinformatics classes based on the .NET Bio add-in ribbon that we see here, and also some additional exercises and tools which we've created for that purpose. The .NET Bio project has an increasingly active community built around bio.codeplex.com, as we can see with the site here. And fundamentally, it's maintaining around 800 downloads per month. So it's becoming an increasingly successful project. Not necessarily used across all of the pharmaceutical industry and big science, but it's getting increasing acceptance from companies such as CBio, the US Food and Drug Administration, Illumina, Amiris, Bionano Genomics, and also from a number of research organisations in a number of different countries, such as the CSIRO in Australia, for example. There are training materials and undergraduate resources available from the site and from members of the community. So what I wanted to talk about today then was some of the applications which we're working on and which others are working on with this library, and some of the future directions in which it might head. I'm going to focus particularly on a thing called Radseek, which is about next-generation sequencing methods for rapid genotyping. So genotyping is a fundamental aspect of the modern sequencing toolkit. It's crucial to any sort of clinical diagnostics based on sequencing. It's also essential for the identification of large-scale infections, particular types of tumours. All of these sorts of things depend on some sort of careful identification of sequence variation. The traditional approaches have been based, and I say the word traditional with a certain sense of irony, but traditional approaches to molecular identification of this sort of variation have been based on large-scale single nucleotide polymorphism chip microarrays. And traditional is, of course, ironic because it's only been around for a number of years, but it's already being superseded by resequencing approaches. The rise of next-generation sequencing means that what was once a ludicrous idea that one could go away and sequence another human genome or another few human genomes is now becoming much, much more feasible. But there are still some limitations. It's not feasible to do or it's not cost effective at this stage to do very large-scale sequencing of human populations in order to identify unbounded genomic variation. That's not the way things are done just yet. We still need to have some sort of selectivity in the samples that are being created, but we want more flexibility than that which is provided by the now traditional methods of SNP chip microarrays. So the problem with those is that microarray designs of that kind depend upon a very good background knowledge of the variation which is being sought. And this, of course, requires a certain substantial prior knowledge of the sequences and their known variability. And that's often a variability and a knowledge which is not actually available to the researcher. So it's important, if we're going to look for genotyping which scales and investigation of large-scale variation across 1,000 or more human genomes, for example, then we need to look at other methods. Now, a number of researchers, notably in Edinburgh and then in the United States, have developed methods based on what they call Radseek, which is restriction site-associated DNA sequencing. And the essential principle of this is to have some use of restriction enzymes to cut out a small section and well-known section or well-constrained section of the DNA sequence, and thence to identify variation based upon that particular sampling. So the alternative here is this use of RAD sequencing on next-generation sequencing platforms. But the idea here is, of course, that you do this for quite a substantial number of samples, generate a large number of reads, and then in some sense collect the reads and identify the variation through clustering and analysis. So there are two versions here which we're going to talk about. Our focus in collaboration with a group at Harvard is based on the double digest Radseek, which we see here. The essential variation is that the double digest Radseek allows you to get greater control over the sample by using restriction enzymes at both ends of the cut. So what you end up with is far less chance of any sort of unwanted selection or unwanted variation of particular cuts. And so by having the use of restriction enzymes, you're at both ends, you're able to control the cut that you're making in a much more refined sort of way, and hence reduce the complexity of the data analytics problem that comes later on. The essential issue here then for data processing and variation that comes in is to group the reads for the individual cuts by sequence similarity into clusters. But we need to ensure some reliability, and we need to ensure that we deal with appropriate throughput. The existing approach for this particular method is based on some cloud storage using Google Docs and some Python scripts. The collaboration that we've got at the moment is utilising the .NET Bio libraries to improve the performance, and also to introduce some new methods for the detection of ploidy, which we'll come along to in a moment. So the essential idea is that we want to ensure the quality of the clusters which are generated, and we want to make sure that we can believe the genetic loci which are identified, and that there's some robustness to the estimates of variation which are put in place. So fundamentally, humans are diploid organisms, and so individuals should not exhibit more than two possible haplotype sequences. And what we're looking at here is some potential variations in the cluster data, which are not in fact the result of the sorts of SNP style variation that we're seeking. These can be due to just straightforward garden variety sequencing error in the clusters where unnecessarily similar sequences are actually grouped together when in fact they should not be. And of course also some contamination from other samples, which is something which one would like to avoid but necessarily arises in laboratory practice. So the focus for us at the moment is on the problem of ploidy filtering. So we need to have some rapid and robust method for detection of ploidy levels within the cluster. Previous approaches, and the one that's being used in the pipeline at the moment, is the use of a simple count and some very simple metrics derived by maximum likelihood estimation based on top of those counts. But other suggestions have included hidden Markov models and discriminative classifiers such as logistic regression or the support vector machine. But at this stage, we're working with a mixture model which has been adapted from previous work which has been used for straightforward SNP detection. So we're using a mixture model with Poisson distributions for each haplotype, parameter estimation via expectation maximization, and the software here is implemented on top of the .NET Bio libraries. And ultimately, of course, the goal is to discriminate between good and bad clusters. So here's this. It's very difficult to make this look as visually impressive as some of the talks that we've had earlier, so I'm afraid I'm just going to have to talk you through what we've got. And the ploidy analysis tool enables the researcher to set up a bunch of quality scores which are essentially put in place as filters to allow the choice of whether these clusters are actually good or bad and whether those clusters should be utilised in fundamental analysis later on. The progress of the run is monitored rather at the right, where we see the number of samples which are detected, the alignment quality, which is a standard quality score measure, and the maximum read quality, which is a measure of the actual quality coming out of the sequencing project. And also we're starting to see what we're seeing in the good clusters which are emerging. And we see the distinction here between all clusters in the middle there and the good clusters at the bottom, which would be the ones that we might decide to keep. The DIRT score that we see there, the DIRT maximum, that's an agglomeration of clusters, or a pool of clusters, if you like, which do not successfully satisfy the requirements of the ploidy, which is associated with the organism. It also illustrates some of the things that we're able to do very, very quickly because of the underlying tool support and the underlying library support. So BAM and SAM file processing, sequence alignment and comparison and management of alignment objects, and of course the management of k-mer objects, which helps us lead towards alignment-free approaches as well. So for a single cluster, just to give you some idea of how this might turn out, you have a particular cluster which has 179,000 reads, which filter down to some 2,138 distinct reads, which correspond to 48 individuals, which was actually the number that we thought were there in the first place. And so on this occasion, the ploidy detection is acceptable, and that cluster is one that we can work with. There's some additional stuff about the read quality scores and so on down the bottom there, which I won't consider at the moment. We see here in the source code the use of the existing bio.dot algorithms and also an additional set of library functions around additional metrics for this sort of work and other next-generation sequencing projects, which has been created by our people as part of this project. So to conclude the talk, some other active projects. So this is the first step in a major pipeline around the DD Radseek, and that's a collaboration that I've mentioned before between us and an evolutionary biology group at Harvard. There's tool support for next-generation sequencing, interfacing to the selectome datasets, which is a set of confirmed positive selection interactions in the evolutionary biology community, and that's driven by Nigel Delaney out of Harvard. Pattern searching languages for .NET Bio, which is a project that we're pursuing at QUT. And we're also just starting to work on a couple of other projects, which I'll mention quickly here. One of the more interesting ones is one that has just started with a student working on a type provider under the F# environment for interfacing .NET Bio and the .NET environment with resources from NCBI. So that's starting slowly with him working on the production of a type provider for GenBank files, but the goal there is to extend that over time to a wider range of Entrez-based resources. One of the more interesting ones, which has got somewhere at this stage, has come from someone in my group who just went away and did it, which was to look at the .NET Bio project and the existing C# .NET source code and to utilise it as a basis for computation and particularly for visualisation support in the browser. And that was the compilation of .NET Bio to JavaScript by taking advantage of a thing called script sharp, which some of you may be aware of. So the idea here was simply to provide a set of bioinformatics objects and some simple bioinformatics algorithms which could actually work within a browser. Obviously, you're not going to do your vast next-generation sequencing pipeline within the browser, but certainly there are times when you wish to do transformations and reformats and simple pattern discovery within the browser when you're visualizing something, and this is really quite an exciting prospect. So to give you some idea, this is the CSharp source code, which utilizes calls to the NCBI web services as supported by .NET Bio, and here is the corresponding JavaScript generated by using C#, and the Bio.js server is something which is a compiled version of the .NET Bio project source code, as I spoke of before. So finally, the project itself resides at bio.codeplex.com. We welcome participation, welcome anyone who wishes to talk about it, and I'm certainly happy to talk about it. And there are a number of other people here who are actively involved in the project, Simon Mercer and others in particular. You can see here that there's also a call out for the use of this project as part of the Imagine Cup. And those of you who are Doctor Who fans may even find this as a way of looking at Matt Smith, the previous or the existing Doctor. So finally, some acknowledgements from Microsoft Research Connections, from Simon Mercer and Rick Benger and all of those people, to my own people at QUT, Lawrence and Amber and various other student contributors, and our collaborators at Harvard, Nigel Delaney, Brant Peterson and Emily Kay, the latter two being from the Hoekstra lab. Thank you very much.
Thank you very much. Very informative talk. Is there any sort of questions or comments?
Thanks, very interesting. I'm just curious, you listed a lot of projects on DNA sequencing and also on protein those. Is there any specific project using this tool for RNA sequencing?
The library itself supports, caters for quite a number of different alphabets. But at the moment, people are not using it for RNA-seq. It's not a major focus of people using the library that I'm aware of. Certainly, if people want to talk about it and are interested, then it's certainly something that people may be interested in collaborating on.
Okay, thanks.
Any other questions or comments? So some of that emerging projects, you are talking about protein structures and so on. So in order to proceed to more sort of translational biology and so on, then you may need a bit more different types of data and images or text, another database. So then the problem may not be just a sort of interoperability of software. That's more data interoperability would become more crucial issues. So is there any kind of a movement towards that direction, like common ontology or some descriptive terms, standardization?
There have been some very basic discussions around metadata, but it's not something which is, people have had their hands full working from what from what's there and adding on things that are very specific to their needs. Mm-hmm. At this stage, people have not looked at trying to extend it towards anything which is really under the sort of the semantic web type of umbrella. Mm-hmm. But nevertheless, it's something which could be supported if there's if people had sufficient interest and a compelling application to make it work for them. At this stage, mostly people are looking for those sorts of services by using the web services hooks to just treat them as external data rather than rewiring the internals of the library to work with them.
Is there any questions or comments? Okay, so thank you very much for the informative talk.